In the beginning of the bactericidal test, the bacteria has been revived from -80° freezer. Here is the name of the bacteria, Lactose garvi. The bacteria were preserved in a cryostant tubes through small beads and are inoculating into the tryptophan soya broth media. The broth media were incubated at 25 degrees centigrade for overnight. The culture media were centrifuged, resuspended in PBS to bring the woody one at 610 nanometer. For the bactericidal test, we require 10 to the power 6 CFU per ml of concentration. So we are taking 9,990 microliter of TSB, tryptophan soya broth, and 10 microliter from the 10 to the power 9 CFU per ml, where OD was 1, and that from there 10 microliter of broth we are taking to make it 10 to the power 6 CFU per ml. Now we are preparing for map, and according to the map we will load different solutions into the 96 well plates. This is our 96 well plates, which is a flat bottom and with lid, and it should be very sterile. In the bactericidal test, each well having 50 microliter of bacteria, 25 microliter of PBS, and 25 microliter of serum samples. First, we are loading 25 microliter of PBS in the wells that contain serum samples. In the bactericidal test, there are two control groups. One is plus bacteria, no serum. Another one is no bacteria, no serum. So in the 4 wells, we are giving 25 microliter plus 25 microliter, that is 50 microliter of PBS in the 4 wells of control group for 25 microliter plus 25, that means the 50 microliter of PBS. In the control group, where there was no bacteria, no serum, we have given 50 microliter of PBS. Now we are giving 50 microliter of TSB plus 50 microliter of TSB, which will contain in a well 100 microliter of PBS and TSB broth. So in the wells, we are giving 25 microliter of serum samples. Already we have given 25 microliters of PBS by the multi-channel. We are giving now 25 microliter of serum samples. We have 20 serum samples we are loading now. After that, we will give 50 microliter of bacterial suspension to every wells. Now we are giving 50 microliter of bacterial suspension, which is 10 to the power 6 CFU per ml to the every wells, but not in the control which having no bacterium, no serum. In this step, we are incubating this plate for 3 hours to allow the bacterial growth as well as to see the serum response against those bacteria. On the principle of serum bactericidal activity, that means the serum having the components of innate immune system, that means the antimicrobial peptides, complement factors, and antibody that can kill or lyse the bacterial cells. Another basic principle is the MTT reduction as a viability indicator. So MTT means the thiazolyl blue tetrazolium bromide, which is we can find in the commercially thiazolyl blue tetrazolium bromide. The quantity is 2 milligram. You can say 2 milligram per ml MTT in PBS. MTT, which is 2 milligram per ml, and this MTT we have prepared earlier, few months earlier, and we can see here these are the MTT, and you can see here yellow color solutions. A basic function of the MTT is MTT dye is reduced to formalin by metabolically active live bacteria. So the amount of formalin formation directly correlates with bacterial viability. The more bacteria lead to higher absorbance and there is an inverse relationship. That means the higher serum bactericidal activity means more bacterial killing. If there is more bacterial killing, then less MTT reduction and lower absorbance. And lower serum bactericidal activity means more viable bacteria, more MTT reductions, and higher absorbance. So these three are the main basic principle of the serum bactericidal activity.
So now you are putting the
MTT.
MTT, and
Yes.
you have to keep 3 hours.
No,
15 minutes.
15 minutes.
15 minutes. Okay. In the last one,
Yes.
nothing should happen because the last is no serum, no bacteria. So the color doesn't change. Here we have, I don't remember, we have bacteria, but we don't have serum.
Serum, yes.
But you see that the bacteria like the serum actually.
Yes.
It's a nutrition.
Yeah.
Are you taking the video?
Yeah, just to how it's changing.
Video is changing, yes. So the color is very strong.
Yes.
But the issue is to find the differences between fish.
Fish.
Can you see them?
Yes.
Yeah.
You can make a video.
Yeah, they're moving a lot.
Anyhow, those who are blue are alive and they're incorporating
The MTT.
and it becomes blue. You can eventually compare the control wells with no bacteria or but nothing.
They did not have any acute reactions.
I know, because the serum itself should contain lysozyme, antibacterial peptides, several molecules which are healing. But in the end, and actually you can see the effect of the serum because in this well we don't have serum. Look at inside, take a picture, you see what happens. So the serum has an effect. Because
Sì, diverso. Serum c'era già.
It is bacteria, no serum.
Janssen, they convert it into insoluble salts, which are purple. You can see from here. Yes.
Yeah, yes.
Compared to the controls. Let's go to the where at 600.
Yeah, 600 nanometer. This is no serum bacteria. These are the wells that contain serum, and we can see the formazine crystals has been formed due to MTT. And formazine crystals are nothing but like salt-like structures and not dissolve in any other solution. So for the proper dissolutions, we have to provide 50 microliter of DMSO and mix several times for the proper dissolution of the pink color formazine crystals. And here we are putting 50 microliter of DMSO in every well.
I will do this way.
And we are pipetting it for proper dissolutions, proper mixing of the formazine crystals so that spectrophotometry will be done in proper conditions. So the web can be transferred through the wells at 600 nanometers.
This is a double, so I can use the same tips, I think.
So we have to ensure for the proper dissolution of the formazine crystals here.
Is it moving
Yeah,
there?
moving. Yes, moving.
Properly with the spectrophotometer in order to be sure that everything is solved.
Once we completed the complete dissolutions of the solution, we'll measure this plate in 600 nanometer in a microplate reader. Yes, now the data will generate 600 nanometer You can see here. Finally, we can see the microplate reader has generated an Excel sheet, but the control, no bacteria, no serum, having the lowest values, and the bacterium but no serum, a little bit higher values. And all the wells of serum are given the higher values. And calculating these data sheets will measure the bactericidal response in the fish immune system.